All right, good morning, everybody. Why don't we go ahead and get started? It's my pleasure this morning to introduce David Stenehjem. Did I get that close?
You got it.
All right. David and I were talking about how I was not going to be able to pronounce his last name. But it's a pleasure to introduce David. He's a St. Cloud State Husky, and then he also has done his PharmD training at the University of Minnesota College of Pharmacy. After that, he went down to the University of Utah, where he did his residency and then his subspecialty training in clinical oncology pharmacy at the Huntsman Institute. He's now the associate professor and associate department head in the Department of Pharmacy Practice and Pharmaceutical Sciences at the College of Pharmacy here at the University of Minnesota. He's actually located up at the Duluth campus. As I said, he's a board-certified clinical oncology pharmacist who specializes in oncology outcomes research, which is going to be part of what he talks about today, and the clinical research in precision medicine in general, something that's near and dear to my heart, and I know many of you in the audience. He's a member of the Masonic Cancer Center, and David's worked closely with myself, Bharat, Mildu, and a number of other colleagues to try and get a molecular tumor board off the ground here at the University of Minnesota. His outcomes research initiatives are focused on understanding the role and value of precision oncology, which I think is something that's incredibly important in this day and age, where there's a lot of hype behind comprehensive genomic profiling, but not necessarily a lot of good data on how much value and clinical utility that actually brings. So with that, I'm very happy to turn it over to David, and thank you very much for coming down.
Great. Thank you. Thank you, Dr. Nelson, for the kind introduction, and it is great to be here on Halloween. So the title of my talk today is Real-World Evidence Studies in Precision Oncology and Harnessing the Electronic Medical Record. For CME, here are my objectives. Let you read them. So by way of introduction, so this is the number of FDA-approved cancer drugs that have been approved per year since the 1950s. And you can see from the 1950s to the 1990s, before the dawn of the biotech era and the genomic era, it was a pretty dismal time to be in oncology, especially in pharmacy, with only about 1 to 2 new drugs approved per year. However, during the biotech boom and then also in the genomic era, we've seen a dramatic increase in the number of new therapeutics that have been approved. And today we're averaging between 10 and 13 new drugs that are approved each year. And 2018 is no slouch itself, and we're, to my count, almost at 10 new drugs that have been approved this year alone. 2016 was a bit of an anomaly with only 3 new drugs, but keep in mind this was also a big year for immune therapy. We had 18 new indications, including many of the immunotherapy indications as well. So dramatic increase in the number of new cancer drugs that we have per year, but also we have a dramatic increase in the price of these drugs as well. So this is Peter Bach's work at MSKCC describing the median and mean cost of drugs from 1965 to 2016, so the same exact time period that I presented in the previous graph. And you can see the logarithmic increase in the cost of these drugs. And today, our therapies are costing up to $10,000 per month for a course of therapy. So extremely expensive new therapies, and some would say in many of these instances, marginal benefits. So really highlighting the need to understand the value and effectiveness of these new drugs in real world populations. So now if we couple in one additional facet into this, and this is now pharmacogenomics. And this is the increase in the pharmacogenomic information that's included in FDA labels by year. And this is up through 2016, and we can see a good increase in the number of drugs that have pharmacogenomic ramifications, included in labeling. And oncology is the leader in this area. And so 34% of these 165 drugs that have pharmacogenomic labels are in hematology or oncology. So now we have precision medicine, high-cost drugs, and a tremendous increase in the number of drugs that we have, which really are highlighting the need to understand how these drugs are being utilized by clinicians and patients. So real-world data and real-world evidence are new terms that have come out. Real-world data relates to a patient's health status and or the delivery of health care that's routinely collected via a variety of different sources. And so typically these sources are claim-style databases. So these are administrative databases where we get information about what was billed to the patient's insurance companies. This is coupled with some diagnostic billing codes, or ICD-9 codes, basic demographic information. And increasingly, what I'll be talking about today is the utilization of electronic health record data to generate real-world data. But also we can think of patient registries, and now increasingly patient-reported data through our smartphones, our wearable devices as well, and how that impacts health and impact on therapies. So real-world evidence now is then the synthesis of that data to form evidence regarding the usage and potential benefits or harms of our therapeutics or interventions, and through the analysis of that real-world data. So there's some challenges to using real-world data, as you could imagine, and the evidence that is generated from them. And I won't be talking about all the pitfalls and challenges, but I'll talk about some that would be near and dear to pathology. And you would think that just identification of cancer cases would be a relatively simple task. And it can be if you have a good institution that has a institutional tumor registry and well-annotated cases. However, that's not always a luxury in many institutions. And even within a tumor registry, oftentimes those tumor registries are lagging behind by several years. And in order to get contemporary data, it can be very difficult to identify cancer cases in a large electronic health record. And a good example of this is an initial study I did in chronic myeloid leukemia. And we used ICD-9 billing codes to try to identify a cohort of patients with CML at the Huntsman Cancer Institute. And by ICD-9 billing codes, we identified 1,000 patients. And we thought we had all these great CML cases. Well, then for an exercise, we then went through and tried to then merge with the tumor registry. And there was approximately 60% misclassification bias by just using those ICD-9 billing codes, which has been commonplace for many of these outcomes-based studies, is to identify patients strictly based on those ICD-9 codes. And then if we couple in then treatment data to the misclassification rate, it goes up even higher. This is actually can be a major methodological challenge, is just identifying cases of interest. Additionally, we have classification issues as well. So staging, I talk about a tumor registry. A tumor registry is the gold standard for staging. However, that provides you stage of diagnosis. It does not tell you anything about when a patient, early stage disease potentially progresses to a metastatic disease. There are some efforts to try to add that information into tumor registries, but that is not mandatory reporting. Without a tumor registry, going into the data to be able to identify progression and so forth is labor intensive and not always as easy as you would think. And then classification increasingly today, based on molecular characteristics or cytogenetic differences is also challenging as well, and how that is integrated into the electronic health system. That same study in CML, we were trying to get cytogenetic results to add into our database. And we were working with ARUP Laboratories, which is a national reference laboratory. It's affiliated with the pathology department at the University of Utah. And the way that they were inputting cytogenetic results into the electronic health record was through a laboratory field that said "see note." And you went to the note, and then it had a hyperlink built into it with a password or a username and password that you had to go in to log in to get that result. And so for the 200 patients that we included in the study, each had an average of 3 cytogenetic results. We had to go in each one of those and type in the password or the username and login for each of those to be able to extract that result and then annotate the note that was included. So oftentimes it's very difficult to actually physically extract this information from the electronic health record to facilitate these types of studies. And then lastly, the assessment of outcomes, as I mentioned, is also can be difficult as well. We are not electronic health records are not set up like a clinical trial database where you have RECIST criteria for monitoring for response. Physicians invariably do this differently within their clinical notes, and it can be a challenge to identify if a patient has progressed or has discontinued treatment for patient preference, adverse events, or what have you. And even survival outcomes. Vital status is also challenging the electronic health record system. Most electronic health records only collect vital status if they die within the institution, or the kind nurse will write a note saying the patient passed away on this date. There are some efforts, especially with tumor registries, to work through the Social Security index data to be able to then repopulate vital status. But even as something as you would think would be kind of our gold standard is relatively difficult to oftentimes ascertain in an electronic health record. So these are some of the methodological challenges with the data for conducting these real-world evidence studies. And I'll talk about some of the ways that we have overcome some of these issues in some examples of the research that we are conducting. So this is a typical setup for electronic health record data. All the data is comprised within an enterprise data warehouse, and then you have data coming from a variety of different sources or different buckets. This can be the hospital electronic health record system, and also a clinic health record system. Some institutions have two different electronic health record interfaces for clinics versus hospitals. And so I know that was the case at the University of Utah. Hospitals used Cerner, where the clinics used Epic. So also posing, methodological issues on how to extract that data and interoperability of that data. We have pharmacy data, the laboratory data, which will be near and dear to your heart, and then the administrative data. And one of the advantages of using electronic health record is that you can get economic data as well. So you can get what is billed to a patient's health insurance company or to themselves. And some institutions will provide you actually with cost data. So you can get a full picture of both the clinical outcomes and economic outcomes as well. So the utility of an electronic health record can be multifaceted. Originally, electronic health records were marketed to improve billing and to improve the fiscal nature of the health system. While there's, today they're billed greater utility for a variety of different stakeholders, for patients and for clinicians. We have improved patient care, and increasingly patients are given access to electronic health records so they can view their own data. But what I'll be talking about is this side of this diagram, the benefit towards researchers, public health, policymakers, and the FDA and other regulatory agencies, and also industry sponsors as well. So for researchers and clinicians, this is where outcomes-based studies kind of reside, but we can also do immunological studies. The policymakers and regulatory agencies are very interested in using this data for post-marketing studies and pharmacovigilance studies. And then also decision makers are increasingly using this data in policy and coverage decisions, and then also in guideline formation as well. Industry, as you can imagine, is very interested in how their therapeutics are being utilized, and this is actually the major funder of the majority of my research. Not only do they do, per se market research, but they're very interested in exploring the benefit of their therapies in different populations, trying to find niches where the therapies maybe work better or should be avoided, and also conducting cost-effectiveness studies. They're responsible for generating that data to give to the decision makers in regards to coverage decisions as well. So I'll talk briefly about electronic health record in oncology because it adds some additional nuances and facets. Because here we have the cancer hospital in addition to the hospital EHR. We have a variety of different specialty services as well that we need to extract data from med-onc, surgical oncology, radiation oncology, and also the imaging information as well. In pharmaceutical data, we have both the infusion center, but we often have that specialty pharmacy data as well that we capture in addition to the standard pharmacies as well. Laboratory data, here very important in these precision oncology studies is giving in-depth pathology information and also outside laboratory information as well from outside vendors per se for companies that do like profiling or any other kind of tests that are being conducted. And as you know, many times it's very difficult to then utilize that information because that comes down to the PDF that gets scanned as an electronic health record, and trying to find that information in this EDW, this lieu of data, can be very difficult to actually track that. And then again, we have the administrative data. So in a contemporary electronic health record, oncology health record, we'd like to connect to a variety of different additional data sources. So here are the tumor registries, but also increasingly at major cancer centers we're connecting to biobanks or biorepositories to understand, to facilitate translational research. So how many patients do we identify? I actually have tumor samples that would be available for translational research. And having an interconnected biobank or biorepository can really facilitate that to have phenotyping of patients, but also then have tumor samples available for these translational research studies. Many states have
have now what's called all-payers claim databases, and Minnesota is one of these states that have one of these repositories. So for an individual patient, depending on, regardless of who their payer is, all the claims information for their healthcare goes into one central repository. In Minnesota and Utah and a variety of other states actually have created these different claims databases that can be accessed. Right now, Minnesota's all-payers claim database is not freely open for access for research purposes outside of the statewide mandates for what they're interested in studying. So an individual researcher like me would have difficulty trying to link up to that data source at this point, but hopefully in the future we'll have a better idea of the comprehensive nature of the care that patients are receiving. As you can imagine, in an electronic health record, you're only receiving data about what care they receive at their institution, not if they go to a competing institution or other outside health system. So we don't need to always receive that data. However, there are increasing efforts to link to other electronic health records as well, to be able to receive that data across a variety of different health systems. And I think we'll see more of that in the future. So we'll have a better idea of the comprehensive care that they're receiving, regardless of what center they go to receive that care. So this is my idea of a contemporary oncology electronic health record and how it's linked to a variety of ancillary data sources. So there are some challenges to using these electronic health records. One of the major issues is structural and with interoperability and incorporation of a variety of different data formats. So in laboratory data, we all know that different reference laboratories have different standard ranges for laboratory values. And that's not oftentimes easy to get your hands on too. And so if you're trying to incorporate data across multiple different electronic health record systems, and this laboratory information is very important, you can have some interoperability issues. Also in clinical, as I mentioned, there's issues with diagnostic coding vocabulary and how that's coded at each individual site. And then also linking departments and continuity of care. So I have examples where we've been trying to work with the surgical reporting system, and that was actually not incorporated into the electronic health record. So all the information that was being conducted during the surgical procedures, including medications and so forth, was not actually coming into the back end of the electronic health record. It was still included in the interface for the EHR, but we could never find that surgical data in the actual electronic health record. And it actually took 3 months of IT time to actually figure out where that data was. So linking between departments can be quite difficult. And then there could be research-specific issues as well. So as I'll highlight in a couple of initial studies that I'll go over, sample size can be a very big issue within a single institution, within one EHR system. And especially in precision oncology, when we're talking about very defined subgroups of individuals, sample size can be a major issue, highlighting the need for multi-site studies. Also, missing data can be extremely difficult. So knowing, did a patient not receive a treatment, or did they receive treatment at a different institution? So this can be a methodological challenge to know about missingness and degree of missingness within the data set. Also, generalizability can be an issue. I've used the term real-world data, real-world evidence, but is the Islamic Cancer Institute real-world data? Oftentimes we use these terminologies, but we're essentially in an ivory tower, and that might not be the exact same care that somebody would receive in rural Minnesota per se. And also generalizability can be based on the populations that are served as well. So in Utah, we have a very homogeneous population where I trained and had my first faculty position. And so we get your comments about the population we probably not generalizable for the rest of the nation. They're probably correct. And then there's also ethical and privacy concerns as well about the sharing and use of this data, especially when you couple in the genomic data. There can be some concerns about how that data is utilized, stored, and utilized for secondary data research. So I'll go into 2 initial studies that highlight the power of electronic health record. And these are both single institution studies that we're using data from the Huntsman Cancer Institute. Both of them highlight the need for multi-site studies, but we'll show the power of using the electronic health record. So the first study I'll talk about is predicted genomic markers in metastatic renal cell carcinoma in predicting response to first-line VEGF TKI therapy. So this was a study that we conducted in And really the purpose was to see if we could identify genomic biomarkers that predicted response to the first-line treatment of VEGF TKIs in this renal cell carcinoma. In this study, the real-world data is not the sequencing data. The real-world data is the outcome. So we actually sent out targeted sequencing to Seattle Genetics that had a 76-gene panel and also included 13 different SNPs as well, and in the study. So sequencing is not necessarily conducted to understand the care. Our primary outcome in this study was progression-free survival on that first-line VEGF TKI therapy, and we correlated that with tumor genomic alterations. And then secondarily, we looked at the mutation profile and what we call long-term responders. So we classified long-term responders as those that had a progression-free survival of at least 18 months, which is significantly longer than the average response to a VEGF TKI, which is 9 to 12 months. And then compared to short-term responders that actually had documented disease progression within 6 months of starting a VEGF TKI. These weren't patients that discontinued due to an adverse event, but they had true progression events. So you can see this is technically challenging to be able to identify a group of individuals that were treated for metastatic renal cell carcinoma, treated with VEGF TKIs, and then correlating that with response and also genomic data. So we were able to identify 82 patients that had first-line treatment with VEGF TKI that had tumor samples available for sequencing. We only needed to exclude 2 that didn't have sufficient tumor, and so we genotyped 80 patients. About 18% of these patients were classified as a long-term responder, and 26% were short-term responders, and then over 50% were in this in-between group. So this is the results of the genomic alterations classified by long-term responders versus short-term responders. And what we identified here is that significant proportion, or trend towards significance, the patients that had an alteration in the von Hippel-Lindau genes were increasingly found in the long-term responders, and only one patient did not have an alteration in VHL, which in comparison, there were several patients in the short-term responders that did not have this alteration. Additionally, we found no patients with a P53 mutation in the long-term responders, which would make sense compared to 3 patients with short-term response. Otherwise, we didn't find any other interesting correlations in long-term response. What's not shown here is the data with FLT1, which is a gene that encodes for VEGF. And this gene is one of the known predictive markers for response to VEGF TKI. And we did see significant considerations with that gene, which is presented here. So with FLT1, this is progression-free survival of these individuals classified by the different SNPs. And so the CC SNP has been associated in the past with inferior response with that first-line VEGF TKI, which we're able to recapitulate here. Granted, relatively small sample size, but there was a trend towards significance here in comparison to the AA and AC alteration. But as you can see with the VHL, the wild-type group had inferior prognosis on that first-line VEGF TKI, and also that TP53 mutated also had inferior response as well. And what we did here is to be able to then group these different alterations into a composite model. So we took patients that had neither of these alterations, whether it was TP53 mutated, whether it was VHL mutated, or either the AA or AC. So these are the alterations that would be what we consider that good prognosis. That was in this 0 group. They had one of these alterations that inferred poor prognosis. They were on this line. And if they had 2 or 3 or more, you can see the pretty abysmal prognosis of these patients on that first line of VEGF TKI. You have a question, too?
So what will those curves look like for patients who were not treated with this drug?
So treated with alternative therapy or no treatment at all? So these patients would all rapidly progress very quickly. So
influence, to what extent can you ascribe this difference to this particular indication as opposed to the tumor biology?
Sure. So this is more of a discovery cohort that we will definitely need to follow up with additional cohorts of study. However, it's tough to tease out at this point whether it is prognostic or predictive. I think what you're leading towards in your question. Most of this, so this is progression-free survival. It's not overall survival. However, what we've also shown in this is that the same trends for overall survival are there as well. They're just not as strong. We don't have as many events in the overall survival analysis. But at this point, what we know that there's data with FLT1 that it is more predictive in nature in terms of in regards to prognostic, whereas P53 is more likely prognostic. But what we're trying to decide here is, in this help us decide which treatment modalities we offer these patients. And patients that have 2 or 3 of these different alterations, we know that they have pretty poor prognosis on this first-line VEGF TKI, potentially should be considered for alternative therapies. We know that they're going to probably progress within 2 to 3 months of treatment on that VEGF TKI, so maybe we should think about alternative treatment. Additionally, this model here has also, in a multivariate model, controlling for the major prognostic criteria in metastatic renal cell carcinoma using IMDC criteria, it still remains significant. So after balancing for the factors that are known to be very important in prognosis for these patients, this is still predictive of progression-free survival in this patient population. I will say this does need to be validated. So the discovery cohort needs to be validated in a much larger cohort of patients, but it does provide some initial data and also does provide some impetus to potentially change therapy or have different treatment decisions for these individuals.
David, can I ask you a quick question to go right into that? How common is the prevalent C allele SNP in the population?
So in this patient population, it was about 10% of patients had that.
Or had the CC, had the homozygous CC, yeah. So that's actually very common.
Yeah. So the next study I'll talk about is our efforts to characterize the response to immune therapy, in this case pembrolizumab, in patients with advanced gastrointestinal malignancies with DNA mismatch repair deficiency. So this was an observational cohort, and we turned this a clinical series of patients with DNA mismatch repair in patients with metastatic GI cancers that were treated with pembrolizumab from 2015 to 2017. This was right around the time that pembrolizumab was FDA approved for this indication, and we wanted to understand what was how was pembrolizumab being utilized in the institution, and we were seeing the same outcomes that were presented at the time of approval. Keep in mind, at this time, pembrolizumab, when it was FDA approved, was approved with probably less than 60 patients total when it went time for labeling decisions. So dramatic response rates, but very few number of patients. So in this case, we used MSI status from pathology reports from ARUP, which was a CLIA-certified laboratory and test. And then we also used radiologists to assess for best response. And so we had a pathology investigator that was included on this study to go through the imaging results for all the studies and do assign RECIST criteria to those individuals. So very small cohort, 13 patients. So remember, MSI is present in about 4% of GI cancer, so actually quite rare. And during this time period, we were identified 13 patients that had DNA mismatch repair and also were treated with pembrolizumab. In this patient population, we had the majority of males, but we did have a variety of different GI cancer types. 50% were colorectal, and we had a handful of patients with a variety of other GI tumors. Majority of patients had at least one previous treatment. And in this case, there were some patients that were treated on an Tumor trial that included pembrolizumab with standard FOLFOX therapy. So this is bordering on, again, kind of more ivory tower, lack of real world, maybe not generalizable for the general population. But these a significant population were treated on that IAT with FOLFOX plus pembrolizumab. So this is the waterfall plot of best response to pembrolizumab in this patient population. The color coding is patients that were treated either with pembrolizumab as single agent in dark blue versus the green color was pembrolizumab with the FOLFOX. We had one patient who was treated with capecitabine. And you can see a very high response rate, 42% response rate, which compares quite favorably to the response rate that was presented in the pivotal study that led to the approval of pembrolizumab in this indication. And we saw responses across the variety of different GI cancers that were observed here. And you can see the majority of patients actually had some clinical benefit from therapy, and we only had a few patients that had outright progression as best response. This is the individual swimmers plot for this cohort as well. You can see that many of these patients are, at the time of analysis, were continuing their response to therapy, so having a very durable response. And again, across the variety of the different cancer types as well, and even some very hard to treat cancers like pancreatic cancer and the cholangiocarcinoma patients as well. They're responding quite favorably to treatment. And also, this is the individual patient data from pancreatic cancer patient that had a complete response to therapy. And you can see the biochemical response by CA19-9. You can see a dramatic decrease, and at the same time, the patient's weight going up as well, and having a profound increase in quality of life and a symptomatic improvement from this disease as well. So we concluded that in our very small cohort, this confirmed to us that this agent does have activity in this disease in patients that harbor DNA mismatch repair, and then again, future studies are needed to really assess the benefit in combination therapy, in particular with the combination chemotherapy. So now I'll talk about how we're trying to improve upon these studies by utilizing multiple different institutions to combine data for these electronic health record studies. And I'll talk about my efforts using ORIEN. And so ORIEN is what's called the Oncology Research Information Exchange Network. And this is a partnership between mid to large size academic cancer centers with a couple different missions. One is to uniformly collect data across these different sites to support translational research and also to promote clinical trial enrollment in precision oncology studies. We're also creating what's called an avatar program where institutions can submit samples for sequencing, and they're actually paid for these samples. So they right now I think their cost, or these institutions are given up to $2,000 per sample for sequencing and the genomic data to be inputted into this database that will be utilized in a more prospective manner. All these patients are actually consented, which is different than many of the other databases like ACRG and so forth, where they actually give consent to these patients. This is through this total cancer care protocol that was started at Moffitt. So these are the current member institutions. I'm still adjunct faculty at the University of Utah and Huntsman Cancer Institute, so I'm getting access to this ORIEN network of cancer centers. You can see some pretty big cancer centers. Moffitt in Ohio are the founding members of ORIEN, but you can see a variety of different centers that are participating here. M2Gen is essentially the for-profit company that is running ORIEN. ORIEN is the group of academic centers. M2Gen is essentially the group that is managing all the different centers and essentially the organizer of ORIEN. And this was spun out of Moffitt in 2006 after a very large bolus of money from the pharmaceutical industry to start creating this data resource. So what I would say is the current capabilities of ORIEN are to be able to conduct these multi-center, both prospective, but also observational studies as well. This is relatively new. There's been a few protocols that have been run through ORIEN, and I'll talk about some of the first those that I've been running in this kind of observational real-world data generation and evidence generation, and we're the first that have been actually utilizing this network to do these types of studies. But they're also in the process of creating this integrated genomic and clinical database, which member institutions will have access to. And this is taking time because, again, it's prospectively enrolling patients, and right now there are up to around 7,000 different cases that have been submitted into the repository. So it's growing pretty rapidly. We have plans to double that in the next year or so, but it is taking time. In addition, in comparison to AACRG, which can go take historical patients, they can go much faster, but this is a different model. What I'll be talking about is not utilizing this database, but creating our own through different research contracts with each individual institution provide data that is not necessarily going into this full database that combines the genomic data. At this point, with the sample size here in these select patient population groups, we wouldn't have any additional benefit for sample size. So we're actually going straight into the electronic health record data to be able to conduct these studies. So these are the 4 current ongoing studies that I have with ORIEN, and I'll tell you about 2 of them. I'll talk about the first 2, so I won't go into depth, but this study right here, we're actually conducting, and Minnesota is participating as a non-ORIEN site. But in this study, we're looking at caregiver burden of patients with caregivers with patients that have GBM. So this is not a true precision oncology study, but it's another study that we're conducting to really assess the burden of this devastating disease. But I'll talk about the first 2 studies that they're doing. So the first one is assessment of tumor mutational burden in lung carcinomas. And this study has been funded by BMS. So as you know, tumor mutational burden is approximately 7% of all cancer cases. And there's a high incidence of this MSI phenotype that also have TMB-high as well. However, that inverse is not true. So only 14.5% of TMB-high is also MSI-high. And this is an increasing group which we're finding that has increased response rates to the immune therapies. And hence that's why BMS is interested in this. And in fact, this is data from Checkmate 227 that was recently published this year. And this is looking at response to combined immune therapy with nivolumab plus ipilimumab versus chemotherapy standard platinum doublet. And you can see in patients with high TMB, so greater than 10 mutations per megabase, this is based on Foundation's medicines, comprehensive genomic profiling, that patients with treated with nivo plus ipi have improved response rates over chemotherapy in the first-line setting for metastatic lung carcinoma. And this is in comparison to here in the low TMB group, where there wasn't nearly the difference between the 2 study arms. So BMS is looking at, how can we redefine the patient population here to identify patients to respond not only with TMB but also with PD-L1 testing as well. So we've worked and proposed this study to BMS to characterize the demographics of patients and characteristics of patients with lung cancer stratified by TMB. And then secondary objectives is to correlate progression-free survival and overall survival and duration of treatment with the treatment with immune checkpoint blockade by TMB, by mutations per megabase in non-small cell lung cancer patients. And here we're being agnostic to the platform. The majority of patients, which I'll show you, have been treated or have been tested with Foundation medicines, comprehensive genomic profiling, about 90% of patients, but we are including patients that have comprehensive genomic profiling other platforms as well to see if there's going to be differences in these response rates based on what platform is utilized. We're also in the study trying to determine the association of TMB with other biomarkers in lung cancer, including PD-L1 expression, MSI-high, EGFR, and other alterations as well, such as POL and so forth. So a very comprehensive approach and taking a big data approach to really assess TMB in this patient population. So currently in this study we've completed a systematic literature review of TMB in this patient population, and we are presenting that data at the SITC next week. And then we have commitment now from 6 ORIEN sites and 5 centers outside of ORIEN to participate, and also sites that have the data capabilities to provide the data to our specifications. And we're beginning the subcontracting with all those different sites. So this is the preliminary counts that we've received from these different institutions. On the top here are the different ORIEN sites that are going to be participating, and down here are the non-ORIEN sites. So here, this is the estimated lung cancer patients that have NGS testing identified at these individual sites from 2012 to present. 2012 is approximately the time where most sites were starting to use NGS testing for clinical practice. And here on the right is the number of patients with NGS reporting with TMB. We also have an agreement with Foundation Medicine that prior to approximately 2015, when they started reporting TMB on their panel for clinical use, they'll actually go back and add in TMB for us for all the samples that were ran prior to that time. So we'll be able to get significant conversion of these patients that have NGS testing over into those that have NGS testing with TMB. But as you can see in these totals, we have a significant number of patients here, over 2,700 patients, approximately 2,700 patients that have TMB reported with lung cancer for this study. So, pretty significant patient population. If you consider, if you go back to this study here, you look at the number of patients that were per study arm, we're talking only a couple hundred. So we'll have some real power to be able to really assess the tumor mutation protein landscape and how that really correlates with response in this patient population. So this is some initial data from Huntsman Cancer Institute. We were able to identify 165 lung cancer patients with TMB at the Institute that were reported. This is some basic demographic information. But this is the scatter plot of TMB by mutations per megabase in this patient population. And based on the cutoff of 10 mutations per megabase, we're seeing approximately 30% of our patients that would be classified as TMB high. Keep in mind, Utah has high rates of clean living, and so we have a much higher rate of non-smoker, non-small cell lung cancer. And in those cases, typically have lower TMB than the cases associated with smoking. So potentially this proportion of patients with a high TMB will actually be increasing the total sample size. So that's a kind of update of where we are to utilize the electronic health record across multiple different institutions to be able to conduct an outcome study, to be able to work with this genomic data and emerging biomarker in the area of lung cancer and other cancers as well. And as for sake of transparency, BMS is also working with us on the same study to recapitulate this in head and neck cancer as well. So the next study I'll talk about is characterization of patients with FLT3 positive AML, and to look at the outcomes and resource utilization in this patient population. And this is due to, this has been funded by Novartis, and as you know, Novartis last year, and FDA approval for midostaurin, which is a FLT3 inhibitor. So their interest in this study was to be able to assess how are current FLT3 patients treated, what are their outcomes, and to be able to prepare for, after we have some real world data with midostaurin, to be able to compare outcomes with midostaurin in the FLT3 mutated group treated with midostaurin versus those that were FLT3 mutated, which really has no other treatment that we specified for that indication. And so really contrast the way that we sold it to them was to be able to try to sell the story similar to the HER2 story in breast cancer, to see if their therapy can make as big an impact in FLT3 mutated AML as trastuzumab did in breast cancer. And I don't think we're comparing apples to apples, but that was the way we pitched this study to Novartis. So these were our objectives, as I mentioned, to compare the historical treatment patterns of FLT3 mutated versus FLT3 Wildtype, and then adding a prospective cohort of patients treated with midostaurin. And that data is currently ongoing, and we need time to be able to generate that data. But this is an observational study, and again, we utilize the ORIEN network and also data from the Huntsman Cancer Institute. And for the sake of time, I won't go into detailed into the selection criteria. However, these were all patients that were treated with induction therapy because in order to receive midostaurin, FDA labeling is to receive midostaurin with induction as NSCLC therapy, and so that was the patient population that we were interested in looking at. So this is the basic demographic information for the Huntsman Cancer Institute. We're in the same process with the ORIEN network, and we have 10 centers that have agreed to participate in this study as well. So we have a really good group of centers, and all ORIEN centers that are participating in this AML study. So this is data from the Huntsman Cancer Institute, and this again highlights, FLT3 mutated AML is approximately 30% of AML, which we see here. The total sample size was 100, nice even 100. And it's like 39 patients with FLT3 mutated AML that were treated with midostaurin. And you can see there's really no difference in basic demographic characteristics between FLT3 mutated or wild type, which is expected. And then, which is expected, is have difference in the risk stratification. And so, having FLT3 mutated puts you directly into this intermediate group based on European guidelines, but no difference in extramedullary presentation or CNS disease presentation between these two patient populations. This is where we start seeing some begin to start looking at the therapy patterns and outcomes. So all these patients received, as I mentioned, induction chemotherapy with 7+3, had very similar times from start of induction to a complete response. This was statistical significance, but it did not meet pure clinical significance. Both were about 14 days, which is standard time to assess the bone marrow. And then complete response rates were actually similar as well. Again, small sample sizes, so we don't have tremendous power to really detect differences here, but at face value, they look at pretty similar responses to induction therapy. However, now we start seeing many differences in the treatment patterns and between the two different groups. So here we see in FLT3 mutated fewer number of consolidation therapies and more therapies for the FLT3 wild type. And what I'll show you is that most of these patients were going to transplant earlier. So they had one or two consolidation cycles, and they go directly into a transplant. And most of these FLT3 wild type patients are getting consolidation therapy and then waiting, they either think they're in remission or they wait until relapse and then get salvage therapy and then go to transplant. So you can see the median days from first to last consolidation therapy is shorter, and then also the complete response is maintained higher here because most of these patients are going to transplant early. So this is the data on stem cell transplant therapy. So median days from index to transplant is shorter here in FLT3 mutated, and the timing of performing the transplant was earlier as well. So most of these patients are going post-consolidation versus a higher percentage going post-salvage therapy in the FLT3 mutated group. And this is data here on the salvage therapy. And so what we identified here too is that patients that with the FLT3 mutated that actually get salvage therapy, the response rates are actually a bit lower than those in the FLT3 wild type group. And that's been shown in other studies as well, that this FLT3 mutated, once they become refractory to treatment, they have very poor outcomes. And we're able to show that as well in this data. And lastly, here are the survival outcomes between these two different groups. So FLT3 mutated here is in this orange color, and there was a trend for worse prognosis, which has been demonstrated before in this patient population. Didn't meet statistical significance, again, sample size considerations. But what we found very interesting is that the number of patients that did not go to transplant, the real divergence in survival outcomes between these groups, highlighting the importance for a transplant in this patient population. And you see a very large hazard ratio here that didn't meet significance. So highlighting the need to try to get these patients to transplant if at all possible. So these data set us up now for being able to overlay in a group that have been treated with midostaurin, and our estimates across the ORIEN network is that we'll have approximately anywhere between 150 to 200 patients in the next year that have been treated with midostaurin, and then also the numbers of patients that the historical cohort of FLT3 mutated group will increase tremendously as well. So we'll have some real true power to be able to see what's the impact of midostaurin in the real world population, supplement the data that's been generated in the pivotal trials. Novartis is also using this information to support post-marketing data as well in the post-transplant setting. So what we've identified in this patient population and through our feasibility surveys to a lot of these member institutions is that we're seeing midostaurin is being utilized post-transplant essentially as a consolidation therapy. And so we're going to be looking at outcomes in that group as well. And we'll be submitting that data to the FDA for consideration and for potential additional clinical trials to support additional labeling claims as well. So to conclude, multi-site observational studies using contemporary VHR can be a powerful tool to understand the impact and value of precision oncology. This does come with some caveats and difficulties and challenges, as I've alluded to, but really you can get high-quality data done correctly with a good data architecture and a systematic way to collect this data from these different institutions. And real-world evidence is increasingly becoming integrated into decision-making from a variety of different stakeholders, from the policy, industry, from regulatory agencies and clinicians themselves for treatment decisions and guideline generation. And these research networks such as ORIEN can really help facilitate these studies to be able to create these integrated data sets, to be able to solve these questions where it can be limited by sample size at a single institution. Also, these research networks can help facilitate contracting, IRB considerations under master protocols that make it just a little bit easier to do our work as well. So With that, I'm happy to take any questions that you may have, and I thank you for the opportunity to present this research today.
Can I start you out with one question on the TMB? This actually came out in one of the recent molecular tumor boards. Specifically with this, from the checkmate trial, the 10 mutation per megabase cutoff. Is that specific to a tumor biopsy or a tumor specimen that was acquired before versus after an exposure to chemotherapy? Does that cutoff change once you've been exposed to cytotoxic chemotherapy?
So we'll be assessing that. One of our data points is timing of the biopsy to see that evolution as well within this patient population. We won't have matched biopsies or two biopsies at different points in time per se, but in the cohort we'll be able to look at temporal trends. Do we see tumor? In 3,000 patients, we'll be able to see. Is there a trend for patients that had that biopsy at later time points after multiple treatments? Does the number of mutations increase? And my hypothesis is that we will actually see that. Some data that I've done in prostate cancer, looking at the evolution of mutations or genomic alterations, shows that the more treatments patients receive, the more alterations you identify, kind of standard paradigm. And so we do expect to see that. In the checkmate trials, these were mandated biopsies prior to treatment because this was a pre-specified cutoff and analysis point that how to stratify those patients. Yep. Questions?
Great talk, thank you. Over the last 40 plus years in the field of kidney transplant, there's been a lot of advances because there has been a national database for kidney transplantation, which was started by Carcinomas in California. And it looks to be like ORIEN might be an initial step to developing a national database in oncology. Do you think it's going to expand to develop a national database in that area?
So that's their goal, is to make a national database, the repository for precision oncology translational research, and also for placement on clinical trials as well, identifying patients. There's other efforts by multiple different groups to be able to create these databases as well.
It's a crowded space.
It is. There is, and so we'll see who bubbles up to the top, and we'll stick around. It's not easy work to do, to be able to try to get all these constituents to play together in the same sandbox is not always an easy task, but hopefully we'll see some that will rise to the top and provide a good resource that would be available to researchers and the clinicians to utilize.
Just on a comment, the people that should have the most interest in this are the payers. And so are the payers involved at all with the analysis of these data? Because, they don't want to spend a lot of money on patients getting expensive drugs that really don't work.
Yeah, absolutely. And so the way it works is that the pharma companies fund the studies that they give to the payers.
Right, but I would just argue that pharma is probably the wrong set of people supporting this type of work.
Yes.
Because in some ways you're talking about finding one that's worthless, and pharma sort of doesn't want to know.
Absolutely. But yes, the payers are very, very interested in utilizing this data, and payers also have access to their own claims data to be able to do their own studies as well, budget impact models and so forth. And so they're getting increasingly sophisticated, and with the advent of combined claims databases with electronic health record data, they're able to do some of these studies. But absolutely, so payers are increasingly utilizing this data.
Whether they're paying for the research to get to there. So that's what I think we need to pitch them at a much higher.
Yeah, I couldn't agree more.
As you know, oncologic pathology is essentially an interpretive branch of science. And pathologists may find various degrees of uncertainty with certain tumors. Not everything is going to be black and white. And you know that many pathologists sometimes use words like suggested of or favor this or favor that. At the patient's level, the oncologists take the pathology report integrated with all the other data and make the best decision possible for this particular patient. I would be interested to hear if you have comments about these research networks adopting diagnoses from different centers with different cultures, different criteria, different pathologist training and whatnot. Did you feel the need for filtering some of these diagnoses, someone to interpret these diagnoses for you, or you did, because I hear presentations like this, I read papers, and I'm always very impressed as to how researchers take pathology diagnosis face value. Every diagnosis you mentioned, GBM, you mentioned appendicitis adenoma, and I suspect this was diagnosed as low-grade mucinous neoplasm. So there's so many there are so many gray zones, and I want to hear your take on taking diagnosis face value.
Yes, so my analogy to this is, so what we talk about in outcomes research is difference between efficacy and effectiveness. So efficacy is a race car going around a closed track, right, taking the highest speeds on a 70-degree day, where effectiveness is that race car trying to get through rush hour traffic in New York City and try to get the same time. It's not going to happen, right? And so in these types of studies, this is important to really provide that real-world data about the effectiveness of our treatments based on this data that is potentially ambiguous, and this is where we see dramatic differences in response rates because of that one of the issues is classification issues. And so, in these studies, we want to understand, all right, when this data is utilized by the clinician, they interpret it, maybe they interpret it wrong, but they still receive the treatment, right? They interpret it one way, and they receive treatment. So what is the response in those patients that potentially had suggested pathology results that weren't definitive? What are the results in that patient population? And so, I'd be interested in working with you to try to highlight that. And we see that all the time in these pathology reports, that it is not cut or dry or black and white, but they're still treated, in the same fashion and same manner. So it's important to try to elucidate what are those outcomes in that patient population.
Here is we basically have one pathologist looking at all of the cases, and one or two people who are trained and send all those samples that we need to them. So
yes.
Kind of standardize it.
Yes, so the kidney cancer example I gave you, we worked with a pathologist in creating our database, reviewing all samples to make sure they clear settle. And we also have done studies in sarcophagoid as well, and kidney cancer may reviewed all the cases to ensure that they have the correct histology, pathology, for those cases. So absolutely, we work very closely with colleagues in pathology to be able to make sure that we're getting the right cohorts of patients. But in some studies, I was alluded to, we want to take all comers and be able to understand the value of these therapies in a broader population. But when we were trying to do more correlative work with biomarkers, we want those very defined patient populations and have as precise diagnosis as possible.
All right, thank you very much.